Right. Well, thanks very much for joining, everyone. We're a couple of minutes over 4, so I think we'll start making a move. So I'm very happy to be able to introduce today Professor Ben Goult. He's come from the University of Liverpool, and we're going to talk about the mechanosignaling properties of a scaffolding protein that you've got a lot of interest in terms of Talin, and we have a lot of interest in terms of its ability to link to lots of proteins that we like. So thank you very much for joining us today, and I'll
Great. Well, thanks, Ian. Thanks, everyone, for coming. Yes, this is actually my first talk with the University of Liverpool logo. I was down at the University of Kent until the end of February, so I'm just in the transition period, spending half my life up there and half my life down in Canterbury. So yeah, thank you for the opportunity. This came about from a presentation which Julia Gorlick saw, which then I gave a small talk at a centre meeting the other week. Gisley was in the audience, got really excited and about the potential for collaboration. And I think hopefully what I'm going to show you today is why that excitement came about and why this protein Talin in the middle of this page is important, and how we're moving it from an adapter, simply a scaffolding molecule, into the mechanical computation machinery inside of the cells. And our most recent idea is this idea of this spatial organization encoded by switch domains in proteins. And that's what I want to try and tell you about today. So the video is going to play, that's good. So we're interested in cell adhesions they make to the extracellular matrix. And that's often shown in this idea of cell migration, as shown by these two fibroblasts in this video from Mark Morgan. This is a cell crawling along an extracellular matrix, and it does so by making these large assemblies shown in green, which are they're often called focal adhesions, but they're integrin adhesion complexes. And this is a very concerted process. It's intentional. That cell's physically crawling, responding to signals at the front of the cell, and then reorientating its whole architecture to pull in that direction. And this is fundamental to the development of multicellularity and for maintaining homeostasis. And it goes wrong in many different diseases, and pretty much every disease you can think of, there can be defects in cell adhesion signaling which can contribute to that. And we're very interested in the immune vascular fibrosis now, blood clotting and cancer. And more recently, and something I'm going to show at the end, the link to childhood epilepsy. And if we look at what that cell's doing, it's physically grabbing onto this extracellular matrix, which you can't see in that because we don't stain for it, but it's like a cargo net of extracellular proteins. And the cell physically grabs onto that. And how it does that is via these integrin, might be ambitious to use a pointer, integrin receptors. So on the surface of all of our cells are these receptors called integrins. And in the simplest sense, and these are like the hands of a cell, because they can physically grab onto the extracellular matrix. And they do so by changing their conformation, like a hand physically grabbing on. And in doing so, that cell can adhere to that matrix cargo net-like structure. And what we're interested in is the machinery which couples that ECM integrin complex to the cell's cytoskeleton. And the main adapter for nearly all of the Talin molecule, all of the integrins, is an adapter protein called Talin. And what Talin also does is it couples the integrin to the cytoskeleton, both the actin, as shown here in yellow, but also the microtubule cytoskeletons. And in doing so, it forms the mechanical linkage between the matrix and the fourth generation machinery. And the canonical view of Talin is that it's an adapter which, when it gets stretched, it recruits a protein called vinculin, which is a cross-linking protein. And in this video in the top corner, that's actually staining for GFP vinculin. And this is, for a long time, this has been what's been considered what these adhesion complexes look like. What's interesting is I'm only showing 4 proteins here: integrin, Talin, vinculin, and actin. But onto that simple scaffold of 4 different proteins, oh, wrong one, the cell can assemble many different structures, which may or may not play. I think it's thinking about it. If they don't, you can imagine that cell we're seeing crawling along. At the bottom, this is a contractile apparatus. This is the muscles of a fly embryo. It's just starting to become contractile. This is going to be a pain. And also, when the cells in our body, they're all held in place via this connections, these adhesions between the cells and their neighbors, but also between the cells and the extracellular matrix. And what we're also very interested in the lab is this coupling between cell division and these two cells, this cell at the bottom, which is becoming 2 daughter cells. At the moment, it's about to undergo mitosis. It lets go of 99% of its adhesions, rounds up into a ball, and then it can split evenly along its towards its poles. You couldn't split a pancake in half very evenly. So it spreads, it rounds up, divides, and then immediately respreads out. So you have to couple whether you're adherent or not to the cell cycle. But all of these structures shown here, and at the end I'm going to talk specifically about the synaptic adhesions, which again are integrin, talin, and actin holding the synapses together. And if you knock out integrin, or you knock out talin, or you knock out actin, you don't have synapses. So this is an intrinsic part of these neuronal linkages. But all of these are mediated via integrin, which is coupled to the cytoskeleton via talin. And we've started working on talin for a good while ago now. I actually started working on this molecule in 2005. And in 2006, I moved to Leicester to work with David Critchley, who was pioneering this talin work. And in 2000, and when we started, we had no idea what this molecule looked like. We knew it was really big. It was 2,500 amino acids. And this rod region, which now looks relatively simple, has got no homology to any other domains. So now we know it's dead easy. But over the course of 3 or 4 years, we worked out what this molecule looks like. And I don't want to go into too much helical bundle detail, but just to say that the N-terminus binds to the integrin in the plasma membrane, and the C-terminus contains a number of actin binding sites, the best with one right at the very C-terminus. And those 62 helices arrange into 13 helical bundles, which we called R1 to R13. Anyone who was interested in talin before this paper in 2013, the naming structure of this was horrendous, but we realized there's 13 domains, so we renamed them R1 to R13. But in this model, you can see that it's bound at one end, it's tethered to the membrane and the integrin, and then the fourth generation machinery is pulling on that, which means that all of these domains are in this force transmission pathway between the mechanical cytoskeleton and the extracellular world. And in that paper in 2013, in the significance statement, we made a comment that talin changes binding partners in response to force-induced conformational change. And that was because we'd found 2 ligands for this R3 domain, one of them which binds on the outside of this switch, and one which only binds when that domain opens up. So hopefully this will play. So one of the things we've done more recently is try and animate what we're talking about, because it's conceptually quite challenging to think about this. But this is a video I made with Sam Barnett a couple of years ago now, although time doesn't feel that long ago, showing what we reported in that first paper. Here we've got the 13 domains of talin, but here we've got one of these switches bound to this BRAP1 effector called RIAM. It sits on the outside. But once we unfold that domain, we expose these 2 red helices, which are vinculin binding sites. So here we can see this is now a textbook example of a mechanosensitive event, how you can convert a mechanical signal into a change in biological signaling. What I haven't got time to talk about today, but something which we published a couple of years ago, was that this is shown in this video, is that the process of stretching one protein can activate another protein, because those vinculins which fly in are auto-inhibited until we stretch talin, and then the exposure of those helices pops them open, and then you make a mechanical linkage, because each of those vinculins will then couple to another actin. So you're building up this network, this meshwork. So in 2013, very interested, how hard do you have to physically pull to cause this switching behavior? And I'll just say as a brief aside, this was work done with Sam to try and visualize this. I think this is probably the paper I'm most proud of, because we present a new idea, which I'm going to talk about later today. All these videos are available on YouTube, and I can send them if you want them. But when I was in 2013, I was a postdoc with Dave Critchley. When we found this switch, that was the pitch for my job talk, that I wanted to work out how these switches work. So in 2014, I moved to Kent, set up my own lab, working just on these binary switches. And at that time, I got in touch with a guy called Yan Ji in Singapore, who was pioneering single molecule biophysics to stretch proteins. And I said, we've discovered these switches. We want to know how hard to pull on them. So that triggered this whole set of experiments and this long-term collaboration, which has moved this whole vista of what's going on at these adhesion sites, in my opinion. And the experiment which we do is we take a protein, a single molecule of Talin. Here we're taking 3 domains of Talin. Oh, wrong way. We can covalently attach them to a cover slip of a microscope via their N terminus, and then we can engineer it so we can attach a magnetic bead to the C terminus. And then we can use a tiny permanent magnet to pull on that bead and exert physiologically relevant forces onto that protein. And when you stretch 3 domains of Talin, what you see is as you pull on it, as you pull this thing taut, it gets stretched longer. But then you see these 3 big jumps in extension. That's these 3 domains opening independently of each other. And what's also shown in this is 6 cycles. So you pull this protein, so it's a complete spaghetti. The second you let go, it immediately resets itself and goes back. So when you pull it again, you see the exact same switch-like behavior. I'm not going to talk about it today, but what we were able to show from looking at the structures, I'm a structural mechanobiologist, is we could identify polar residues inside the cores of some of these domains, which make them more mechanically sensitive. And the R3 domain is the most mechanically sensitive domain in Talin, and that's the domain in that video just now. And it takes 5 piconewtons of force to open that first domain in this system. And since then, we got much better at the cloning. So since then, we got much better at the cloning, and we were able to then do a lot more proteomics on Talin. And what we start to see is that these 13 switches are binding to many different proteins, and there's over 40 different ligands for Talin, and this is very abridged. If we repeat that single molecule experiment, stretching from R1 to R13, this is the data which we get, which is still absolutely mind-blowing, even though it's like from 2016. This is the behavior, the mechanical response of a single molecule of Talin. And what you see when you look at this is you still see the R3 domain open first, which was in that video at the start. But then what you see is this stepping stone, step-like behavior as each of these domains overcome their own force threshold and unfold. And because each of these domains are binding to ligands on the outside and the inside, and we haven't even identified them all, these steps each represent a single molecule changing its mechanosensing and changing its biological signaling in response to force. So we think that this mechanosensing at these sites is incredibly complicated and sophisticated. Something else which we discovered in that paper, I've actually cut out the slides where we've actually exploited this, but what we could see is if you pull on a molecule really hard, then these domains open, pop, 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 pop. But that introduces a lot of slack into the system. So you can pull on this and the tension on the molecule won't ever get above 10 piconewtons up until half a micron in length. And this is the cell's shock absorbing system, because if the cell gets a bash, the whole thing doesn't rip off because these domains will pop, pop, pop and open. So this is a cellular shock absorber. And I cut the slides out, but we've since made this into a material which has got shock absorbing property. So half of my lab is currently funded by the Ministry of Defence, because we're trying to make this into a new type of armour because of its ability to stop hypersonic projectiles. But that's off what we're talking about today. What we start to do, and in collaboration with Martin Schwartz in Yale, who's been very helpful for me in developing this as a theoretical framework for understanding mechanical signaling, we started to build this framework of this notion that Talin's serving as a mechanical signaling hub, and that in a certain environment, when these domains are all folded, you recruit a certain set of ligands, and because these are 2-state switches, they're either open or closed. they're binary switches. So we started to call them 0 if they're folded, 1 if they're open. So in a certain switch pattern, I've lost my cursor. You've got certain switches, and you can call them 0, 0, 0, 0 for simplicity. But the readout is the enzymes and the molecules which are recruited in response to that pattern. And if you get a mechanical signal which causes an unfolding event, then the weakest domain in that series will open next. And that will change this pattern. Here it's changed it to 0, 1, 0, 0. And the readout's changed because that red guy, I don't know what colour that is, has been displaced. And you've recruited a cytoskeletal protein, probably vinculin. But as I'll talk about later, there's enzymes which also come in. And when you pull on this again, then the next weakest domain unfolds. So you're constantly changing the signaling at that site as a function of these quantized states, of these switch patterns. And this led to us writing one of my favorite type of review articles, one of these at a glances, a couple of years ago now. And if, as a blatant sales plug, we just got a re-up of these where JCS did another print run because I'd sent these out. If anyone wants one, then I've got a whole box of them now. But if we look at this and start to imagine what this molecule is doing, then we've got all of these different domains which are opening and closing and coordinating different signals. This isn't a bridge list because the ones we're currently working on we haven't put on here. But you start to imagine that this is quite complicated and sophisticated at the bottom of our cells because talin's connecting to the integrins which are connecting to the different components of the matrix. And in doing so, it's interrogating different mechanical environments outside the cell. But then it also couples them to different cytoskeletal connections, both directly through direct actin binding, but also indirectly via vinculin coupling to integrin. And it also is the main coordinator of the microtubule cytoskeleton at the cortex, because there's a protein which I'm not going to talk about today called CANK, which forms the foundations for the microtubule stabilizing complex at the cell cortex. And we can show that that's true because you can put a single point mutant in one of these domains and microtubules no longer target to cell adhesion. They just grow out everywhere, totally random. So this is Talin integrating the two cytoskeletal structures in response to mechanical signals. But what it means is this is super complicated because we've got a network of all of these competitive interactions with these switches which all open and close independently, and then all the different ways that this molecule is experiencing tension, and both from internal and external signals. And that means that there has to be some coding going on here. This is robust. It happens in all development from the first fertilized egg. The whole mechanical computation of the cell works out as part of the organism. And this is also probably why it goes wrong in later on. If the mechanical inputs get corrupted, if it starts to get fibrotic, if it starts to get stiffened, if you start to lose this coupling, then this whole machinery can start to missignal just as well as it maintains homeostasis. It can start to adopt and optimize against the wrong conditions. But what this is all I've talked about so far is a single Talin molecule. This isn't what's going on. There's probably 100 Talin molecules in an adhesion. And what we've also more recently shown is that vinculin, which binds from Talin to actin, is also got 5, maybe 6 switches, depending on the rate you pull. So you've got this complex meshwork of switches on the bottom of our cell, all of our cells, all animal cells. And then in response to the mechanical signals, the history of what's happened to that cell, these will be in specific switch patterns. And then they recruit all of these different enzymes and signaling molecules and adapter molecules and RO and RAC into those adhesions in a dose-dependent manner, depending on the status of that signaling machine. So what we start to think is that Talin is a mechanosensor, that's well established. And in the context of mechanotransduction, in the context of a migrating cell, you could start to imagine that the switch patterns which form are kind of like a balance between the extracellular forces and the cell's force generation machinery. And the switch pattern depends on how many signals from either side. So you build specific signaling complexes as a function of that, and that dictates the signaling outcomes. But something which changed my thinking of this and changed how I thought about this was this very simple question: is what happens if your cell's migrating along a surface, it doesn't know what the ECM is going to, what the next bit of ECM is going to grab onto is going to be like, how stiff. You've got two variables in this scenario. But if the extracellular environment becomes stable and predictable, and that doesn't mean it doesn't change, the cells invest a huge amount of resource to remodel that ECM. Then here you've got a different proposition. You've got a series of switches connected to a predictable surface, which the cell's force generation machinery can operate. So this led to a change in my thinking about what mechanical signaling might look like. And it also led to this idea that this is imbues mechanical memory functions, because each time you pull on this, each time you switch on the motor proteins of the cell, you pull, and in doing so, you change these patterns, and then you change the signaling. So this led to me just at the start of lockdown writing, like everyone did, actually finishing their papers. I was just flying back from Australia, sat in Sydney Airport, had this idea, oh, if it was stable, then it would be different. And it led to me writing this paper. And this paper starts out by laying this context of mechanomemory and mechanical memory in proteins, which is what we can physically measure. We can do this experimentally, and we can manipulate these switch patterns experimentally. But then it took it one step further to say, if the organism has organized all its cells so they're all operating to this same mechanical coding, then you can start to write information into these, and you can change the signaling and the behavior of the cells using small changes in contractility. Small pull on the motors, you update this pattern. So this led to this big idea, like the brain might be a mechanical computer. This was probably too much isolation, but this was like the big idea. This is like, these switches are absolutely essential for synapses. If we knock out Talin, we haven't got a synapse. But then this patterns would be updated by the contraction changes as the signaling occurs, and that would change the activity of those synapses. I'll talk about this more in a second, about why we think this might be true, because the big question is how. And what it led to was the idea, could we build a simulation of this? Could we actually see this 10 mechanical cues leads to this output? And then we realized that's way too difficult because we're not computational modelers. But something we realized as we were building the components of that was that something which was obvious in all of that single molecule data, but we'd not really appreciated, was that these molecules, when those switches open, they're not just recruiting different molecules, they're also changing their dimensions in one dimension on the micron scale. Like this protein has been measured at 0.5 micron in cells. Like physically, the coupling of a GFP at one end, cherry at the other end, it can stretch up to 0.5 micron in a migrating cell. And that's because as these domains open up, they start to unpack a huge amount of spaghetti, a domain going from a folded bundle to 150 amino acids of intrinsically disordered or spaghetti protein is a big difference. It goes from 5 nanometers to 50 nanometers. It's a 10 times increase in length, to the point that a single switch unfolding essentially doubles the length of this molecule. And that leads to this, that there's these figures, which everyone will have seen, maybe the top one, maybe no one wants to see my poor equivalent. It's massively distorted in the Z dimension. If you look in the supplementary of Tony's paper, you see it yourself, the talins are much longer. It averages down to this length, which just happens to be the length of fully folded talin. But in reality, this is just one snapshot of what these adhesions look like. And it's very similar to the Mercator projection map of the Earth, because this is super useful. Everyone's talked about this for the last decade. Everyone's got this common framework for telling stories, for understanding biology, but it's incredibly distorted in the Z dimension. And it's like the Mercator projection map of the Earth, because there, where Greenland looks the same size as the whole of Africa, because the Z is compressed, which is very useful if you're trying to navigate, and everyone knows it, but it's a huge distortion in one dimension. Because if we redraw those complexes and those structures and those what adhesion looks like, then you start to see that you've suddenly got this separation in one dimension. Because each of these proteins, each talin is attached to a specific attachment site on the integrin, and then it's getting pulled by the contractile operation in one dimension. And here we're just showing 3 different domains opening up. But what I'm also showing on here is I'm putting on the ligands for these different domains, because then we can start to see what's happening, that these spatially resolve along Z over to 100, 200 nanometers. And these distances are encoded via the switches. And what we're focusing on is we're focusing on the enzymes and the enzyme substrates which bind to this, because you've got this idea where if the enzyme and the substrate are really close together, you could have high activity. But if you physically hold them far apart, then they never see each other. So the effective concentration goes almost to zero. So you can dial up and down in enzymatic processes physically by just moving molecules relative to each other. And we're working, I'm not going to go through all of this because that'd be tedious, but we're working on, I want to go for the PKA, the CDK1, which is what I showed in the video at the start with the mitosis, and also this ATAP one, this acetyltransferase. There's three examples of how you can integrate mechanical signals into chemical enzymatic processes. And I want to go through this quite fast because I want to get, but this paper we published, it came out like 2 weeks ago now in PNAS. This was a fantastic collaboration with a guy called Alan Howe and his student Mingyu Yazumi, who's in my lab, and then Yan Yu, who's in G's lab, where we discovered that Talin contains ACAP helix, which is a helix which recruits the protein kinase A machinery to those sites. And this is exciting for me as a Talin guy because this is the first enzyme which binds on the inside of a switch. Bit of a spoiler for the next few slides. But PKA does everything. It's everywhere. It does all this stuff. And how it works is it's specifically targeted to regions within the cell. This is well established now, and you guys work at it, so it's by. But how that tends to happen is that the regulatory region of PKA binds to a specific helix. It's called an ACAP helix, A kinase anchoring protein helix. If that helix is exposed, then a PKA will dock onto it with high affinity, nanomolar affinity. And then it will exert very localized effects because it will be targeted to that specific site. And those ACAP are single helices, and Talin's made of the helices, there's 62 of them in the rod. And I'm not going to go through this in much detail, but needless to say that PKA is active at focal adhesions. It's changing the morphology, how many there are. And if you make, if you deroof a cell and look, then PKA is at the adhesion sites and it pulls down in the focal adhesion fraction. And if you make a FET sensor for PKA activity and target it to focal adhesion sites using the Paxillin motif, then you can see that the adhesions light up. The PKA is active at these focal adhesion sites. What that tells us is that there had to be some form of anchoring site there to recruit this protein. So the idea was to find what that was. So using a bioID assay where you make a PKA regulatory domain with a mini turbo promiscuous biotinylase, you then target it and then you see which proteins get modified. When we do that, what you see is that Talin is modified by it's biotinylated in that assay. I'm not going to show that detail because I want to show it at the protein level. But the good thing about the ACAP field is there's a beautiful assay which has been set up by others where you can run any protein you want onto a gel, and then you can essentially do a Western blot, but instead of using an antibody, you use the PK ACAP binding site, and then you use the secondary antibody to visualize it. So the nice thing here is you can run it on, the helices can reform in a gel, and then you can see which proteins can bind to your enzyme. And the other nice thing about this is you can compete this out using an ACAP helix in solution. So you can run this in the presence of a competitive inhibitor to show that this is specific. So it's beautifully simple. Whoever worked this out, is hats off to them. It's fantastic. Talin's a really big protein. So to get at this, the pipeline which we've worked out is to divide it into smaller chunks. And then whichever chunk is a positive signal, divide that into smaller domains. And what we do when you see that is that only the R9 to R12 region, those four switches, binds, and it can be outcompeted by a competitor helix. So that was straightforward. We knew it was one of those four domains. So then the obvious thing is you just start chopping off a domain and domain and domain, and you run this same assay. And what this clearly showed was that R9 to R10 will light up really nicely, but the R9 domain doesn't. So this told us that it must be R10. But the curveball came with R10 is also terrible on its own. So somehow both R9 and R10 are required to get this really strong signal, but neither's really sufficient, which was a bit of a problem because it was like this is one helix, so what's going on here? And luckily, as an NMR spectroscopist, and this is a salesperson for NMR, I'd say it's absolutely the best tool for visualizing what's going on in these things in an assay-based format, because we could physically see what's going on. And if you take R9 and you label it, this is, I'm not going to show you too much NMR, so don't panic, but this is an NMR spectra of R9 in grey, and then it's an NMR spectra of R9 plus pKa in blue. They overlay perfectly, so they don't interact, exactly what we saw in the overlay assay. And the same with the R10 domain, they don't interact. However, once you have R9 and R10 together, then all of a sudden half of the signals disappear from the spectra. So that tells us that when you've got R9 and R10, you've got a big unfolding event which takes place. And again, we know what every single one of these spots corresponds to. So we know which residue it corresponds to. Here, I've just coloured them blue dots, or is that purple, for the R10 and orange for the R9. If you look at this on that other plot, then what you see is all of the ones which are from R10 are still fully present, but all of the signals from R9 have disappeared. So that tells us that R9 pKa unfolds R9, but only when it's attached to R10. And it doesn't alter the structure of R10 because the red dots overlays perfectly. So this was a bit of a curveball. What it turns out is that the very last helix of R9 is an ACAP helix, but it requires a tiny amount of R10. And it's only about 2 amino acids of R10, actually, as I'll show in a second. But then we can use canonical ACAP disrupting mutations, these AD mutants on that helix, and we can kill this interaction dead. So we can actually use this. The problem is, it kills the Talin pKa interaction, but the reason why this is mechanically gated is that those residues are normally buried inside the R9 domain. So when we mutate them to aspartates, then that domain's unfolded as well. So that means that we constitutively activate R9, which is a limit. So the new generation of ACAP mutants we're trying to make are to try and not unfold the domain. And we're quite confident that this will be doable, because if you look at how much of R10 you actually need, again, it's a very simple assay. If we have the full helix of R10 here, then we can unfold R9, and then we just make it shorter and shorter and shorter until it no longer can open up this domain. So now we can physically see you need about 10, but it's actually nearer to 4 amino acids. You need a tiny bit. So the motif involves the linker region between the two. But this is adding in 400 micromolar of pKa. So it sort of unfolds this domain. If you look at this at the single molecule level, going back to our stretching experiments, then here we're looking at 4 unfolding steps of Talin for the R9 to R12 region. And this is so robust, this protein, because we can do this all day long, and we'll always see those 4 unfolding steps. So there, R9, R10, R11, and R12. If we repeat that experiment, but add in pKa, and we're only adding in 10 nanomolar of pKa in here, then on the first unfolding step in the presence of pKa, you still see those 4 unfolding steps. But after every subsequent unfolding event after that, you only see 3 unfolding steps. So pKa doesn't, at 10 nanomolar, pKa doesn't bind to Talin. But once you stretched it one time, pKa will sit on that and it will limit the refolding of that R9 domain. So this is the first mechanically gated ACAP. We had a bit of a heated discussion with the editor that Esrin is as well, but in our opinion, it's not, because it's constitutively exposed. But it's mechanically gated, and you can see that as well, because if you have blebistatin into cells, then you lose that biotinylation of Talin. So again, to try and simplify this, we make a video to just illustrate what this looks like, and that's shown here. This is Talin coupled to integrin and actin. Once this R9 domain unfolds, that blue helix, which is the ACAP helix, is exposed, and then it recruits the pKa at that specific site. And it's quite a specific site because it's from the integrin, the length of that tether up to that point. So it's along the force vector. And this is what the summary, a video summary of our paper. It means we can update our table here of what else is binding to this system. And it gives us a new way to try and look at how mechanics and signaling and computation might be occurring. Now, I don't want to go through the CDK1 interaction, but I just want to show this one video because this is a summary of our paper from a couple of years ago. CDK1 sits on the R8 domain of Talin, but it also phosphorylates Talin. This is summarizing 4 years of work in a 15-second video, but it phosphorylates Talin. The phosphorylation of Talin doesn't change its interactions or very subtly, but what it does do is it destabilizes R7 and R8, which is a 9-helix module, which means that the next time we pull on this, that's the next weakest domain in this chain, so it unfolds. The consequence of unfolding 9 helices is over 100 nanometers change in length. So pKa is sat on this domain when it opens up. So 1 minute it's somewhere up here. If CDK1 modifies Talin, then all of a sudden the pKa is targeted still to this site, but 100 nanometers further into the cell. So here we've got two enzymes which are talking to each other in a sort of allosteric way, but by not ever actually touching each other. They're touching the scaffold, which is then changing the position of both. But it leads to this logic gate operations argument, because if you don't have the phosphorylation, if CDK1 is not active, then that's a strong domain, so it doesn't unfold. But if CDK1 is active, it phosphorylates Talin, then the next mechanical signal will pull and that domain will open. So you can have and or kinase and force or whatever. So this adds basic computational rules, I would argue. And what's so exciting to us is that things like GEF-H1 and other substrates are phosphorylated by these two enzymes, and the phosphorylation differentially changes the activity. Here we can imagine that they're targeted to the focal adhesions via the microtubules. They come into close proximity and then they get edited depending on the status of this machinery. So we're very into this now, and we're trying to work out ways to measure this. Is that enzymatic activity will be coordinated by spatial positioning. The big difference here is that this spatial positioning we're talking about is 50 nanometers, 100 nanometers, 150 nanometers. They're small changes which change the effective concentration in many, many orders of magnitude. And it's not hard to imagine that if you do bring the enzyme and substrate close together, you've got high activity. If you hold them far apart, you've got low activity. So this is what that tells us is that Talin's signaling a scaffold protein controlling the flow of cellular information, exactly like in MAP kinase cascade and in downstream of growth factor receptors. The big difference here is that this is mechanically operated. These switches can tune this, dial this up or down. So something which we wrote last week was a bit more of a perspective on this. Well, we didn't write it last week, but it came out last week. To try and define this concept of mechanically operated signaling scaffolds. And what are relevant to this discussion here, or this meeting here, is that this is very sensitive to the extracellular environment and the amount of contractility of that cell. And if you do change if the cells are fully in control of the ECM and it's built perfectly like a nest, it's completely known to the cells, then this mechanical signaling happens with great fidelity. If the environment's changing, either during development or during disease onset, then the stiffening is going to be changing this, and then the signaling is going to start to get out of kilter. So this is where this collaboration starts to come together. We can physically start to look at this because you guys have worked, or Gisli's team are working on TGF-beta and integrin driving fibrosis. And this mechanical signaling is downstream of TGF-beta and integrin. And what you start to do is as you stiffen the extracellular matrix, then you change this mechanical signaling. And because Talin's regulating ECM deposition, it looks like you've got this feedback loop where stiffening is driving more stiffening. And this is all workable, and it might all be like modulatable by understanding this switch-like behavior. But what I want to spend the last, I don't know how long I've got left, the last 5 minutes on, it's something which is, I think, leads to a bigger idea about what's going on here. And it came from this simple question, is here we just drew these nice balls of 30 nanometers in radius because they look pretty, and you get this nice Venn diagram effect. What actually determines the size of those spheres? So something else which we realized as we started to model this is that Talin is long. I hope I've convinced you of that. But the molecules which interact with Talin are also pretty long. And they have this standard arrangement where they've got a binding site for Talin, a big bit of spaghetti, and then the enzymatic domain. And that means that the enzyme gets bolted onto this scaffolding, and then it can operate anywhere within a certain sphere, which we call the zone of activity. And this is actually our model of what we think ATAP1 is looking like. It's binding via a single helix to one of these domains. And then that means that its enzymatic domain at the other end has high activity anywhere within this sphere, because it's tethered, so it can't get outside the sphere very well without letting go. But any substrate within that sphere will be processed by this. And what you can immediately imagine is if another domain unfolds here, this whole machinery is going to move 50 nanometers towards me, or reciprocal if a domain unfolds at this end. I don't know, because it doesn't even show me. Then it'll move to the membrane. So this is spatial positioning of an enzymatic activity, which we think is very similar to this guy and this goat in a field. If you stake a goat in a field, you'll get a perfect circle of tightly mown eating grass, which will be a perfect circle around the stake point, the length of the tether. If you move that stake somewhere else, it'll have access to another set of grass. And if you make the tether, the linker longer, it'll eat a bigger circle of grass. And that's what we think is going on here with these enzymes. And again, we can measure this, draw these molecules to scale. And then here's some of them with their zones of activity. And then we can start to see what these look like inside cellular compartments, all drawn to scale. And the thing which we focused on was, because we're very interested in this binary coding, was what this would look like inside of a dendritic spine, because that was, it's a very nice discrete compartment. I think these are perfect mechanical systems because they're totally isolated from the cell body. They're just out there, a perfect sealed mechanical unit. So we think that this is how you can have mechanical computation. But if you look at what a spine looks like, it's blatantly a cell adhesion thing, because it starts off like a filopodia, it reinforces and it builds. It's exactly the same as how you make it, how a cell migrates. It sends out these protrusions, then grows them. It's just here the whole cell body is not able to go with it. If you draw this to scale, then what Talin looks like is, it's a bit hard to see, but Talin is shown fully closed in A and then fully open at the side. As these, and that's with the all 11 vinculin binding site, 11 vinculins engaged. The dimensions of Talin span the entire length of these systems. And if you take these zones of activity and then lay them onto their respective binding sites, you see what I think is going on here, where the spatial organization of these enzymes and substrates relative to each other sets the activation of that system, to the point that you could have some patterns where enzymes and substrates are perfectly lined up. So you've got this perfect sequence of events. Or you can have some patterns where these enzymes and substrates aren't. They're frustrated because they can't meet the right substrate. So you could dial up and down the activation of a system by positioning of the enzymes in that thing, in that as a function of switch patterns. And that's exactly what we're starting to see. Sorry about the quality of this. I think it's your screen rather than my slides, but I do apologize as well. But if you start to look at this using antibodies with the N and the C terminus of Talin within spines, and this is work done by Tony Koleski and Rob Nisha at Yale, then what you see is that the green dot is always at the end, it's always at the top of the spine, and then the pink dots at different lengths away from it. So we don't yet show that each, so we can measure, we're trying to triangulate this by having a third colour in the middle. And what we really want to do is correlate these lengths, which we can measure with the activity of those spines. But it's promising that Talin is changing shape. It's that Talin is in the spines, but also that it's changing shape in different spines within the same dendrite. But it leads to this idea of how enzymatic processes are coordinated via this binary coding, because you're spatially organizing these molecules in one dimension, because all of the integrins at the top, all of the contractilities at the bottom. So you're pulling this up and down like this. And it means that you could have 4 spines which look identical. This is speculative, but this is what we're trying to prove. But the enzymes are positioned differently as a function of these signals. And each time a synapse fires, receives a signal, the motors get switched on. That's not our work, that's what's established. The contractility, the cytoskeleton remodels, it changes the positioning of all of these different enzymes and substrates. And we kind of broadly say it's a bit like a gearbox, which works very nicely in the UK. Terrible, and if you go in America and try and talk about gearboxes, because they don't have gear sticks. But in our cars, or like a gear stick, what happens is you use force to move one part of the machinery relative to the other part of the machinery and fix that so that the transmission is set differently. That's what we're really desperate to try and prove, that these binary switches are set in the enzymes in all of these linkages in such a way that information is sent around the brain networks differently by setting the weight of each synaptic connection. And then each time there's a signal, you update that one. And that's why these are so beautiful as mechanical units, because if one gets signaled, the other ones don't change because they're completely isolated. So I've got one more slide, I think, of highly speculative. But if you can imagine that you can write information into proteins, into the shape in a binary format, and that this could set the activation of enzymes, then you could start to imagine you could write information into this scaffolding in all of the cells in our body. But in particular, the cortex of the brain, which is where information is thought to reside, it's incredibly repetitive. They try and say it's not, but it is. It's like 2 million of these columns of neurons, and they're all talking to each other. They're all wired up. If you take any one of these teardrops, they can have up to 60,000 connections to others. This is an incredibly complex wiring diagram where all of these electrochemical connections are set up. And in this model, what I'm trying to propose is that the scaffolding information could be written into the scaffolding of the cortex in a way that sets where the electrochemical signaling flows through that by setting the weight of each of those connections, exactly the same as in a solid state disk. Because then all you need to do is give a specific address in memory in the solid state disk. We just say channel 1, NAND flash 6 or whatever. And then as long as you address that information, you can just have it there and just call it back whenever you want. And I think that's exactly what's going on in the brain. This is the big idea we're trying to prove. But then you can start to imagine that every single thing you do starts to write into that coding, that this is a mathematical problem we're talking about, where you've got a binary coding. And this guy going for a shower, you put your hand in, it receives signals as receptors. If it's really hot, there's a reflex action. But information sent through his neurons, through his spinal cord up to his brain, and then a calculation occurs, and then it sends the right input back down. And this is just going for a shower, but you could imagine every single stimuli which we ever receive updates the coding. And that's what we're trying to prove, is that there's a mathematical representation of an animal which happens from in utero or in egg or wherever they come from, and then is updated throughout that life to optimize it for its own existence. So just to summarize, because I want to ground it back, because that's wacky, that's like the big idea. But this is trying to prove this, and proving mechanical computation has led to profound insights into how cells are sensing and responding to forces, how they're migrating and how it goes wrong in diseases. And the concept of mechanical memory isn't pie in the sky. We can physically take a Talin molecule and we can write information into it, and we can read that information out by different, what we're starting to do is read that information out by having different fluorescent proteins which recognize the different 1 and 0 states. So we've got whole intrabodies for this whole molecule now. Well, we span the whole molecule, we've got 5 of them which work. But ultimately, we want to be able to read the shape of these molecules, because we think the shape of this molecule is what is the information in cells. And because this is running in every single cell, we think that this is a mechanical synchronization of all of the cells. They're all linked and they're all coupled at different levels and different degrees, but they all run in synchrony until they start to spiral out of control. And like aging or like disease, fibrosis, that starts to change certain regions, and then they start to missignal. And in the brain, I think that this is, as I've said, I think this is where information is written. Memories, engrams are written, physically written, in the shape of the scaffold molecules in each of these synapses. But the nice thing here is this is completely consistent with what everyone looks at and the current thinking of memory, where electrochemical signaling is controlling it all. The only difference here is that this is actually the computational step, which is under the hood, which sets the flow of information, which synapses have got high activity, at the minute you just read the probability of vesicular release, but that's like who knows why that is. But what I also think is that this identifies an addressable read-write mechanism, memory system. And just as a final note, anyone, it doesn't matter what your idea of memory is, it's got to have an addressable read-write memory system to form any sort of computation as we know it. You've got to be able to store information, act on it, and then generate the right output. And I think that that's what this identifies. And it supports the view of information processing systems obeying similar rules and logic, obviously in a different format, but to enable computation. So I'll finish there. I just want to thank, in particular, Ji and Ming Chi. I emailed them in 2013 just to stretch a single molecule. I had no idea then how profoundly different our view of this would be. And every single paper we do and every single project, the ones we're writing at the minute, completely changes how we visualize this molecule, how we visualize mechanotransduction. And I'd like to thank Sam because he made the nice videos with me and helped me model this type of stuff, which I think has been very instructive. But then a special thanks to Martin and Nick, because they help with the theory. They're big critics of it, to be honest. But every time they criticize it, it kind of makes it better because they can't debunk it, they can't break it. So that's a good sign of that we're on the right line, and the experiments seem to be leading in that direction. The guys who fund this, and just to finish, if anyone is looking for a postdoc, then I've got this was in December. I said, oh, I'm just about to advertise a postdoc. Totally misunderstood how bad university administration is. It took 4 months. This went live last week, 4 months after I sent that initial message. But if anyone would like to come and work on this, in particular in mechanical imprinting in cardiac disease progression, then please drop me an email. So thank you all for your attention.
Thanks very much, Ben. Some fascinating ideas, very huge impacts, and some nice pictures as well. I'm going to ask a couple of questions first while we get some more from online. A lot of the images that you showed and the concepts you've spoken about, not all the time, but a lot of them, they're talking about kind of Talin as a single molecule, but you know that it exists in a huge mesh with all these other Talins and all the other things. So have you found evidence of them not necessarily always working as a sort of singular kind of conduit of information, like when one extends, it is having an influence on the extension of one's next?
No, that's a good question. That's also like, because the other problem you run into if you're going to have memory written in the shape of molecules is if one molecule pops off and gets degraded, is that information gone? And it's like, no, we don't think it is, because of exactly that. Like, it's an ensemble of all of those switches in that signaling complex. And the idea came originally from thinking of stable outside, but also that there's gaps and gaps and things with opposing functions bound to Talin. So if one Talin gets stretched and then increases contractility, there's a whole feedback loop because of the gaps and the gaps which are like balancing. So I think there's a stable state to whatever this computation is, where at that state the switches are in a certain pattern across the Talins in the complex coordinates that. So yes, and the other big challenge here is most of this was done stretching end to end. When the vinculin binds there or microtubules are attached, they don't really pull, but they exert a big drag on this. Then there's orthogonal forces as well, and they could have very specific, but the end product is the same, that you end up with a certain switch pattern. So yes.
And this switch pattern came from the idea of one thing being constant.
Yes.
Where you want it. Can you take these Talins or from different sort of diseases or different ages or something like this and see whether it's differently? So if you took, say, a young person or a young animal model's Talins and put them into an aged system, do they suddenly have more of a young kind of?
That's so. The hard thing is when you let go of this, it resets itself.
So it does. It always resets because
apart from inside these domains, there's serines and threonines which get phosphorylated, and that's where the new project came from, because we were looking at healthy heart and unearthed cut, and the modification, the post-translational markings of Talin change over time.
Because that's what you were sort of saying with your idea, is that these modify things so that this is part of the learning of the system over time.
Yeah.
So, but you're also thinking that they can be completely reset, or
yes.
Once you've learned a certain amount of things, the epigenetic kind of, well, not epigenetic, but the phosphorylation that occurs on these different residues can be sort of undone again, or they're now sort of imprinted so that you can put them in a different system.
I think both. I think, and especially because we've only talked about the kinases here, but there's also phosphatases at this end. So you've basically got a molecule or molecules which are getting phosphorylated, dephosphorylated, depending on their spatial localization. And those patterns might be like, one thing I don't think that my adhesions in my hands are constantly turning over all the time here at this precise moment. I think there's a stable solution to it where in the tissue, these things are quite long-lived and these cytoskeletal structures are pretty stable. I think in a cell in culture, because it never reaches mechanical homeostasis, they're constantly treadmilling, they're making assembly, disassembly, which has tricked tricked everyone into thinking that these are incredibly dynamic structures. I think they're not that dynamic. I think that as soon as they rebuild and they constantly, but not on the time scale which we see in cells. So I think there's the lifetime of these, it's hard to say for sure.
There's one on the chat. Yeah, go.
Yes, so this is from Scott. This is a fascinating talk, thank you. Cytoplasm is obviously a complicated colloid gel, so Talin isn't moving to a fluid. Talin enzymes may not have sphere activity. Does that create additional fine-tuning, stepwise directionalities?
Yeah, it's a good question. And the colloid, like the sphere, this is, and the other thing to really stress here is this is the maximal size, because that's when it's fully extended, which is thermodynamically not that favorable. So somewhere between that sphere is going to be where that enzyme is going to be positioned. And also the other thing which we sometimes get asked is that the angle of the adhesion is not straight up all the time. Maybe under the nucleus it is at this angle, but it doesn't make any difference because the enzymes are still positioned along a single line. So yes, that will definitely contribute, but it will also be a known contribution because the cell will have to manage that complexity, that colloidal sort of nature itself for all its functions anyhow. I don't know if that answers it, but
Kevin, did you have a...
Yeah. They were really quick questions. Now you've identified Talin as a mechanical gated ACAP. So you reckon there will be other ACAPs that's also mechanically gated? And the follow-up question would be, is there a uncoupled state of Talin without coupling to aliens?
So whether there's other mechanically gated ACAPs, I'd imagine there could well be, yeah. There's lots of proteins. It wouldn't surprise me at all if in like the link complex or at the cell-cell junction, there's also cryptic ACAPs which get exposed when the tension loads. If you let go of all of Talin and it's not got anything else grabbing onto it, the domains refold and then it folds up into a tight form, an auto-inhibited state. So in much of it in the cytoplasm is all wrapped up on itself, auto-inhibited. So it unpacks at the membrane and then once it couples to integrin and actin, then force starts loading on it then. So part one, I think, yeah, there probably is other mechanically gated ACAPs. And part two, it rolls up into a ball.
It's clearly a fascinating molecule. Thank you very much. I know very much about it, like everything. When Talin report forms, and we refer to this with the recap data a little bit.
Yes.
All those proteins are firmly attached. Yes, so they have to be actively ejected to allow Talin to be formed.
Yes.
Let's talk through what the mechanism of that is.
So vinculin's the canonical unfolded rod binder, because when one of those helices gets exposed, vinculin binds onto it. If you have constitutively active vinculin, it will bind and it will never let go. That's lethal in all animals, and the cells just get completely stuck because the whole machinery gets clogged up. The really smart thing is that vinculin has also got an auto-inhibitory state. So vinculin, so if we stretch a Talin molecule with constitutive vinculin, it'll bind, never let go. You'll never get that refolding step. But if you do the same experiment with full-length vinculin, because vinculin can also auto-inhibit, when you lower the force, Talin's trying to auto-inhibit itself, that can auto-inhibit. So then it actually comes on and off in quite a nice, more orderly fashion. For the enzymes, most of those enzymes, apart from the pKa, which is very new, then we're still trying to work that out, they bind on the outside. So when that domain unfolds, it's kicked off. The other bit which I've not talked about here is there's quite a lot of competition for some of those domains are quite hotspotty where there's multiple ligands which will compete. When and where pKa gets kicked off, yeah, I don't know at this precise moment. It might be that once it's on there, it's pretty stable and that domain. Because the other interesting thing is that the domain which pKa binds to when it's open, R9, is the main auto-inhibitory domain of Talin. So pKa also stops Talin folding up and auto-inhibiting itself. So it's probably having a massive allosteric effect on both adhesion dynamics just by stopping auto-inhibition as well as phosphorylation. It's at all of the connections between the myotendinous junctions, and it's also at the, I don't know actually, yeah. So where they meet the two. So because there's a thin layer of ECM at that point. But whether it's at other places, I don't know. It would definitely be at the neuromuscular junctions as well. It'll be at the third line and at the junction with them,
Yeah, very fascinating. Now, I have a couple of questions. I don't know how clever they are, but when I understand the expanding of the protein will be energy dependent, the contracting will be energy free, will be like the favorite entropy energy, minimum energy when contract.
Yeah.
Okay, so it's free of energy, totally free. The contraction.
If we make these proteins in solution, they're always folded.
They're always folded.
Yeah, we don't see them folded.
And another question that I have: what are the modifications that this protein have with age? Which sections exactly have to damage?
Yeah, I don't know whether the protein itself ages, but the changing. So on the note of the refolding, one of the things why this has got memory is that it takes, if you stretch a domain, 15 piconewtons, say, unfold. They don't refold when you go back down to 14 piconewtons. You've basically got to go down to 3 or 4 piconewtons. They've got this huge hysteresis effect. So once you have a temporary increase in tension, you unfold a domain. All the while it's under basal tension, it won't refold. And we could hold a molecule at 5 piconewtons and none of the domains would ever refold over the lifetime. So the hysteresis is important. Mm. Whether this molecule itself ages or whether they're turned over every now and then, I don't know. But the mechanical conditions which make the mechanical system, like the ECM changes, the connections to the other parts of the cell, they start to get less perfect.
And in muscles like the heart, for example, the contraction, expansion is no controlled by the nervous systems, by it's happening all because all over the continent.
Yeah, so one of the things is that's where the grant came from, because yes, these post-translational modifications change over disease state, but they also become imprinted so that, like the phosphorylations, to stabilize this system. I don't think it's constantly every single heartbeat. I don't think having to do this, because I think it's actually stabilized to a certain pattern with the certain enzymes, and then it's just maintained. Then if you do slightly more exercise or work, then you pull it and then you change it. So I think that one of the big aims of our grant is to actually get a real deep dive into the post-translational modifications of this. And
in the case of the heart, what would be the controller of the span of contraction?
Well, this is the attachments where the two cardiomyocytes are held together, or bits of here. So everything will be pulling from that. So when the heart contracts, in the simplest idea, when the cells are contracting, it would be pulling against the two ends. And then somehow, like, because it's not just, there's many different mechanosensitive bits in the heart. It's just these are the ones which are physically holding it together. And this is an interesting story. Yeah. That would be, yes. Good to know that. Yeah, just definitely look at that.
And in the case of the lump, we do the circulation of the part. As we said,
Yeah.
would be like a partial expansion is so.
Yeah, I don't know how much that's another ought to be worked out as well, like the tissue level changes in contraction. Attachment size, whether these become they're changing on the same cyclical pattern, I don't know is the short answer, but you'd imagine that they would, but as long as it's doing it reproducibly or at the same within the same normal range, you're probably not in doing this all the time. But then if you have a bigger exertion or bigger change, then it might then change again from that.
And can be a situation that this system is locked, that it cannot all contract or expand.
Yes, if they are modified so they can't refold, then if tension does go down, it will get shorter, but it won't go refold back up. So they will still be open, but they'll just be like linked with profit. But then they'll get pulled out on the next cycle. So I'd imagine that that's the big idea of the grant, to actually try and show that this becomes what's the dynamics of this at different levels of organisation. So something on that, and we might relate to, I don't know what the guy's name is there, is that one of the things we wanted to do was actually to prove that mechanical homeostasis is at work in the animals. So one of the things which we did with Martin Schwartz, which may or may not come out in Vision at Science Advances, I don't know whether they'll but hopefully they will. But we put a single point mutant in between the R1 and R2 domains. And then if you look at the ascending and descending aorta, then what you see is that you can see profound differences in the collagen organization around I forget which one, because the two population stem cells grow towards each other, and on the either above or below that joint, you've got less tightly organized collagen. So just by tilting the mechanical homeostasis, we can change there's still way more to work out. I think we've just found this computational device in the cells, and now we need to work out what it does.
Couple of quick questions. Sorry if you addressed it. If you adjust the extracellular matrix and do these experiments, what's the effect on the folding? I mean, presumably it'll have an effect. You have to use more force to get it to the bottom. For example, when you do these experiments, you're doing them, they're all computational. You haven't done anything of, say, tissue culture plastic and then matrices of 15 newtons or 50 newtons.
So there's the classic experiment with Dennis Disher and Adam Engler where they put stem cells on different stiffness of ECM. And if you put it onto jelly, they basically differentiate into fat cells. You put them on glass, they differentiate into bone cells, or look more bone cell-like, I suppose, more accurate. So that cell's physically interrogating its environment. So one of the things which we're really keen to do is to actually look at that in the context of the switching behavior and whether you could actually, so what the kicker I would look, the best experiment I'd like to do, if I can get someone who's good at stem cells who would be up for it, is to put mutations in Talin which soften domains such that it feels like it's on a hard substrate and then get it to differentiate into a fat cell on glass. Because Talin's at the top of all of the mechanical transduction as far as I'm concerned. I think people will argue with me vigorously about that, but to me, you've got the integrins got by the ECM, but the first responder is the Talin just inside the cell.
Okay, and what about if you subject cells to cyclical mechanical structures?
Yes.
Cells that are dynamic.
Yes.
Cardiomyocytes of cells. How does that affect? Do they open and close?
Yeah, so that's why we're making these interbodies to actually try and physically read that. We know that the length changes from using a GFP cherry and watching as it moves. You can see them get longer and shorter like that, cyclical contraction and extensions. But what we really want to see is to see like 3 or 4 points along it and try and visualize that and try and show that when and where. And that the problem is that there's no the reagents didn't exist. It's why we've made these antibodies, which are specific to the domains.
And would it be like a, like a or springs? If you pull it within the circle, subject it to certain forces, it will go back. But there'll be a point at which one thing is force and can't.
Yeah.
And any idea what sort of level of force requirement? Presumably that injury could do that. Could you do reversibly screw up Talin?
Yeah. And also, if you pull too hard and it had the brakes, if it lets go of the actin or it decouples, then that will start to reset, reprocessing. There's a lot of enzymes which are cutting this, like calpains, like so they might be. Yeah, it's really complicated. I don't know. Most of these questions I don't know the answer to, but that's because it's still to be worked out.
Yeah.
Yeah. But one of the most exciting, so the memory bit, which is probably the wackiest bit, the most exciting thing there is that Talin binds to APP and CDK5. So APP is the main cause of Alzheimer's, and CDK5 is the main cause of childhood epilepsy. And we've got a whole series of mutations in the actin binding site of Talin-1, which cause childhood epilepsy as well. So we're trying to publish that with Wendy Chung in Boston Children's Hospital, because there we've got a very specific neurophenotype in patients, which is caused by mutations in this gene. And there the idea would be that the enzymes get out of sync to the point that you get a reentry circuit and the whole thing goes. So yeah, watch the space. But I think the amount of diseases which are going to turn out to be on this once it's classified as a disease-related gene, that's what I talked about this morning. It seems a long time ago now. I think it's going to be massive because I think it's coordinating every single cell in the process in some respect.
Any questions? Well, so the amount of overlap with stuff that we know is already involved in IPF, ACAPs, the APA1 integrated biology, so the amount of overlap. One other mechanical process we need to be involved in IPF process is spindle assembly and cell cycle control. Is there any role of Talin in those kind of processes?
Well, definitely the CDK1 binding, because one thing is it's modifying the adhesion, but the other thing is it's also depressed. Like, so I think that CDK1 is stabilizing adhesions during S phase. Well, it is because they get bigger during S phase, but it's also involved in stabilizing cell-cell junctions. So this balance of cytoskeleton architecture, the cell and cell matrix, is coordinated by a CDK1, and Talin is doing that at the bottom end, and there's other things which do it on the side. So I think, yeah, there's a link to the cell cycle, and it's a big challenge because if you go from a single cell to an organism, you've got to tightly couple your cell cycle to the whole thing. So I think we've got a CIEK program grant with Martin Humphreys and Tash Yorkson in Manchester to look at this, because the link between mechanical signaling and the cell cycle goes badly wrong in disease. And in pancreatic cancer, where you get a lot of fibrosis, they get much more aggressive phenotype. So the changing stiffness changes this whole system. So maybe these diseases are a good way to get at the normal healthy behavior as well.
Well, thanks very much, Ben. I think I think lots of brilliant work to come and lots of collaborations as well.
I think so, yeah, that'd be great.